Welcome to ISL-enabled Virtual Lab of CSIR.
Crop improvement is a priority for India and many other countries. We all want crops that yield better, that can resist diseases better, or be tolerant towards floods and droughts. Creating these plants is one thing, and then having these plants reliably pass on these characteristics from one generation to another is quite another thing. And it has been a long-standing puzzle. Apomixis is a technique that scientists say is going to help in doing this. Today, we have Professor Venkateshan Sundareshan with us, an expert of Apomixis, to discuss what this really is all about. Welcome, Professor Sundareshan, for joining us today in this conversation. We want to hear from you about Apomixis. What is Apomixis? What kinds of work has been happening in your lab in this direction, if you can tell us?
Thank you for having me do this interview. So Apomixis is a name of a natural process that many plants, about 300 different plant species or more undergo, that is in terms of reproduction. So normally a plant will reproduce by making pollen that then fertilizes the egg cell within the ovules and make seeds. In apomictic plants, they just start making seeds right away, quite often, the word apomixis itself is from Greek. I think it means apo means not, and mixis means mixing. So they don't mix genetic backgrounds. It's just the daughters are identical, or the seeds are identical genetically to the parent, and this continues for many generations. And some apomixis are very common. There's a plant called the dandelion, which is not so much in India, but in Europe and North America it's apomictic. It's a weed actually, but it's been quite successful genetically. And but the main thing is that all the plants are clones of each other, genetically speaking. They have the identical DNA sequence. And my lab actually didn't start out working on apomixis. In fact, we were researching how does a sexual plant reproduce. And we worked on rice because for technical reasons. So it turned out that what we learned from rice for sexual reproduction could be manipulated to make rice reproduce asexually, like an apomictic plant.
And which means that if you have a certain characteristic in that plant, it will keep on continuing.
Yes, exactly. So the traits are transmitted over generations, and normally speaking, in sexual reproduction, the traits will segregate. This is something discovered by Gregor Mendel about 2 centuries ago, and but in these apomictic plants, the same traits are carried on generation after generation. And there are practical implications to this, that if you have a combination of traits that you want to keep in one plant, then this is one way to do it, especially when they make a hybrid plant. And hybrids in most crops are much better in terms of yield compared to inbred lines. It's called hybrid vigor. Heterosis, the common term. But then that's only for one generation. The next generation, everything segregates. Thanks to Mendel's laws, that everything falls apart. But if you are apomictic, that hybrid vigor can be generation after generation. So right now, farmers have to buy seeds if they want to plant hybrids. They have to keep buying hybrid seeds and replanting. And seed companies also spend a lot of money making hybrids. But if you could do this through apomixis,
then you get that.
Then you make it once and then it self-perpetuates.
And how do you make these apomictic plants in your labs?
So what we discovered through basic science, again we are studying sexual reproduction, is that there's a couple of genes, and most importantly one called baby boom, and for reasons that may become clear. And this gene is expressed in the from the male side, okay, and it's silent in the female side. So the egg cell keeps its gene shut off, and the sperm cell actually is ready to turn it on. When you bring them together, then embryogenesis starts. So we thought that maybe this gene is a trigger or a key for starting embryogenesis. So what we did is to change the expression of this gene. Now it's switched on in the egg cell. Normally the egg cell switches it off, because normally the sexual plant wants to be fertilized. Yeah. But if you can switch it on, then the sexual plant starts making seed right away.
Oh, wonderful.
Without making, so now it's asexual.
Yeah. There are already these tools of genetic modification that we use in agriculture. How does Apomixis enrich that? Or do you see that Apomixis has already reached a level where it can be used with the other crop improvement tools that we have for agriculture?
So Apomixis itself would be a crop improvement tool. Right. It relies on, but to do it you have to use certain basic molecular tools, like gene editing, CRISPR-Cas9, which I'm sure you're familiar with, developed by an institute, by Dr. Jennifer Doudna and Emmanuel Carpentier. And Jennifer Doudna's institute actually funded our initial work. Okay. That is, she started this institute called IGI in Berkeley, India. So it's very much a part of the toolbox, molecular toolbox, that's already being used. And all we did is we had to eliminate some genes that are required for segregation. They're called meiotic genes. And we had to switch on baby boom, the rice copy of baby boom, not a transgene of. So essentially this will operate on top of existing crops. So you find a good hybrid with all the properties that are desirable, and then you make it apomictic by making these gene edited modifications.
Right, but we still don't have apomictic rice out there in market, or it's far away from that.
Yeah, so, and with any of these things we have to do field trials first. That we are just starting. So.
Okay. So basically, are we just this field trial away from this reaching the market, or there are other requirements?
There are probably regulatory requirements which are,
but not technical.
When we do the field trial, we'll find out. So, but it is ready to go to that stage, and in theory, everything should work.
Okay, it's actually great that your work anyway was in rice, and this. Rice is a staple crop, and now having this modification in rice already makes it like a usable thing for people. But do you also see this working out in other crops?
Yes. So we published a paper showing that baby boom will also work in maize copy of baby boom. And I think in all cereals, because if it's in rice and maize, it's going to be all. Outside of that, we're still experimenting. But in all the cereal crops are, after all, a major staple for in all countries, whether it's rice or corn or sorghum or whatever. Yeah.
Right. And so these are all cereals. And if people want to make non-cereal kind of things, I mean, I would imagine that the baby boom gene would also work equally well in other.
So we are still doing tests with that. At the moment, we haven't gotten that kind of efficiencies and nobody has. We've been trying, but I think in plants that are divided, flowering plants are divided into monocots, which is cereals, and dicots, which is all the legumes, for example, and vegetables. So in those crops, it has not really worked out. But probably some of the same components will be there. Certainly for the meiotic genes, it looks like the same components will be there. But it's less fundamental research has been done in these dicots in terms of what makes a zygote or what happens after fertilization.
What makes a zygote of a dicot different from that of a monocot?
Yeah, or in fact what makes a zygote work in a dicot. The basic research is the foundation of our work, and that similar work has not been done as exhaustively in dicots yet.
Wonderful. So that's it that we wanted to discuss today, but we understand that the field trials are on for the rice apomictic
Well,
crops.
they are in progress, yes. Yeah.
And hopefully the work required for the dicots as well takes up pace, and
Yeah.
we'll hear more of that soon too.
Thank you. Yeah.
Thank you very much.
Thank you for having me.